All right, so I think we're going to get started, hopefully what you gleaned out of the presentation prior to lunch is that one of the commonly used techniques in cancer biology, and of course it's not exclusive to cancer biology, is the manipulation of DNA and RNA so that you can change gene expression, you can overexpress genes, or you can try and dampen gene expression. This is commonly done now by using something called siRNA that Lou Dory talked about. Of course, what you're hoping is going to happen when you manipulate the DNA or RNA to change gene expression is that you're actually going to change the expression of the protein that's encoded by the gene that you've been trying to manipulate. So what I'm going to talk about is just two commonly used techniques to look at protein expression. Oops, sorry, I didn't. Okay. All right, so the two things that I am going to talk about, as indicated on the slide, are immunoblotting, or also called Western blotting, which is a technique that's used to detect proteins in cell or tissue extracts. The other technique I'm going to discuss is immunohistochemistry, which you use to detect proteins of interest in fixed or frozen tissue sections. It's much more commonly used in fixed tissue sections, and I'll describe what I mean by a fixed tissue section. But both of these methodologies, what they have in common is that they rely on having an antibody to the protein of interest in order to detect the protein. Okay. So starting with the immunoblotting, there's three essential steps involved in immunoblotting. And the first step is to actually separate the proteins on a polyacrylamide gel. So what you do is you take your extract, you usually heat it in a buffer that contains SDS, which is a detergent, that it will coat the protein, it will unfold the protein, so the protein has no more secondary structure, it's completely unfolded, and it renders the protein negatively charged because the SDS is negatively charged and it coats the protein. So once you have your extracts and SDS, and you have a gel that's poured, this is a polyacrylamide gel that also has SDS in it, and you have little wells in the gel, and you can load your protein samples into the wells. Usually you also load a marker, which is proteins which are different colors where you know what the molecular weights are. And so all of the proteins will migrate through the gel towards the anode because they're negatively charged, and they will separate solely on the basis of molecular weight. You can vary the concentration of the polyacrylamide to resolve proteins depending on what size proteins you're interested in. You can vary the resolution of the protein by varying the concentration of the polyacrylamide. Okay, so once the proteins are separated on the polyacrylamide gel, then you want to transfer the proteins from the polyacrylamide gel onto a membrane so that you can then basically immobilize the proteins on the membrane and then further process the proteins as I'm going to describe to you. And this, generally speaking, is just done by an electrophoretic transfer. So you just sandwich the gel, well, you actually place on a membrane, either nitrocellulose or it can be this PVDF, it's a polyvinyl-derived membrane, and you can just put the gel on top of the membrane, put filter paper soaked in buffer on top and the bottom, and put it into an apparatus, again, that just allows, there's an anode towards which all of the proteins will flow, and then they're just transferred from the gel onto the nitrocellulose membrane. And now we can further manipulate it. So there you have right here. So what they're showing you here is now you have all of the proteins. These are your markers, colored markers, and then you have your proteins, which you can't see yet, but they've been transferred and immobilized on this membrane. Okay, so finally you come to the protein detection step, and this is where you need the antibody. So you have your membrane, which is here, depicted right here with your proteins immobilized on it. And you take an antibody that specifically recognizes the protein of interest. This is a critical step, that you have an antibody that's specific to the protein that you want and doesn't recognize other proteins. But sometimes that's not always the case, but that ideally you have an antibody that's specific to the protein of interest. So you've incubated the membrane with your primary antibody. Before that, you've blocked all nonspecific binding sites on the membrane by incubating with something like dried milk with milk protein or BSA or some other protein that blocks nonspecific binding. So now you incubate with your antibody. Now you come in with a second antibody, which will recognize the primary antibody. And generally speaking, you use a second antibody from a species different from the antibody from which the primary antibody is derived. And the second antibody is species specific. For example, if you used a mouse antibody, your primary antibody was a mouse antibody, then you come in with a goat anti-mouse or a rabbit anti-mouse secondary antibody. And that antibody is tagged with an enzyme. And what the enzyme does is it catalyzes a reaction which either produces a colored product or it produces light. And if it produces light, you can then just, after you add the substrate, you can just expose the membrane to film, and it will wherever on the membrane light is being produced, it'll expose the film, and you can detect the position of the protein that way. Yes?
How do the proteins refold after they've been transferred?
They don't refold, actually. They're probably still denatured. So the antibody has to recognize an epitope in the unfolded protein.
Another question.
Yeah.
What's the advantage of this over an ELISA assay, which also would use antibodies?
Well, in an ELISA, you have, I have done an ELISA myself, but you have, I think the antibody is immobilized, actually, on it's a way to measure the protein by binding to an immobilized antibody. So this is actually telling you what the molecular weight of the proteins are, and you can see, well, I'll show you some examples where you can actually.
But in ELISA, you might need two good antibodies also, because one to capture it, one to detect it.
Right.
And if you don't have two good ones, you can't do that.
So I'm going to show you actually some, just as an example of the kind of data that you might see in the literature. I've borrowed a figure from a paper by Mary Hendricks, who's actually a PSOC team member. And she's interested in nodal signaling. But the thing to notice on, well, let's just go through the various lanes. Whoops. Sorry. Okay. Yeah.
Why do we need two antibodies?
Two antibodies to detect. Well, first of all, I mean, it's very hard to get a primary antibody to have every antibody tagged with an enzyme. So what you do if you have a secondary antibody that recognizes all primary antibodies from a certain species, you can just take that antibody and use it for multiple types of Western blots in your lab. You don't have to tag every primary antibody. So number two, it amplifies actually the signal to have a secondary antibody, because I think it can bind at multiple sites in the primary antibody. So it amplifies your signal, and it prevents you from having to tag every primary antibody that you're using. That's right, yeah, no, okay. So I haven't done this for a while, so okay. So what the Hendricks lab has found is that certain cancer cells are expressing a protein called nodal. So if you look, these are actually separate Western blots, but let's see what, okay. So these are 3 human embryonic cell lines. So these are cell lines which are pluripotent, and they're able to differentiate in to a lot of different types of cells. That's one of the characteristics of stem cells, is that they can differentiate into a lot of different cell types. Each one of these lines that she looked at is expressing this protein called nodal. And nodal is actually contributes to the pluripotency of these human embryonic stem cells. Now, this is a melanoma cell line right here. Now, you can see that's expressing nodal, and this MB231 is a breast cancer cell line, and what she's showing on this Western blot is that is also expressing nodal. But this is a normal melanocyte, not expressing nodal, nor are two normal cell types from breast, two normal breast cell types. Okay, so nodal expression, at least in this experiment, seems to be confined to the two cancer cell lines that she looked at. Okay, now it turns out that she also examined in a separate Western blot the expression of a protein called lefty. Lefty is also made by human embryonic stem cells, and it antagonizes nodal signaling. And you can see here, actually, what you can see on this Western blot is that there's 3 bands. They all represent lefty proteins because the protein is produced as a precursor and gets processed in the cell to lower molecular weight types. Okay. What she noticed is that in the tumor lines, the melanoma line and the breast cancer line, lefty was not being produced. So these cells were making nodal, but they were not making a protein that antagonizes nodal signaling. And she was able to show that if she took the lefty protein, which is actually a secreted product, secreted from the human embryonic stem cells, and added it back to the melanoma and breast cancer cell lines, that she was able to actually mitigate the aggressiveness of these cancer cell lines. So this paper actually created quite a splash, but this, of course, was reliant on being able to measure the expression of these proteins by using Western blot. Okay. All right. So now we're just going to turn briefly to immunohistochemistry. So this is a method to detect proteins, your proteins of interest, in a fixed tissue, not in an extract. And again, it's the same principle as what you use to detect proteins in immunoblotting, in that there's a primary, use a primary, oops, I keep hitting the wrong thing. Okay. So you use a primary antibody to your protein of interest. The protein can be either intracellular, or some of them are cell surface, the proteins on the surface of the cell. You come in with a second antibody that recognizes the primary antibody, amplifies the signal, and that's tagged. And generally speaking here, you're using it's tagged with an enzyme that catalyzes a reaction that produces a colored product. Now, before you're adding the antibody, what you're doing is excising your tissue of interest, and you're incubating it in formalin, and what formalin does is it cross-links all the macromolecules. It kind of immobilizes the protein inside the cell, but it preserves the tissue architecture. So you're able to see where the protein is in the cell, and if the protein is in the cell, which cells in the tissue is the protein expressed in, because often when you'll look at proteins of interest in a piece of tumor tissue, you will find that the proteins are not expressed uniformly throughout the tissue. That's because cancers are often not homogeneous. They're usually heterogeneous. So even within an individual tumor, you will have cells that are producing certain proteins and cells that are not producing certain proteins. This is one of the big problems in treating tumors, is that tumors are not homogeneous. They're usually heterogeneous because of the genomic instability, which is constantly at work in the tumor. So you fix the protein, you fix the tissue in formalin, and then you embed it in usually in a wax called paraffin that enables you to take the tissue block in the paraffin and slice it into very thin slices, which you need to do so that you can actually visualize the tissue. And then you just take the slices and you mount them on slides, and then you go through this procedure where sometimes you have to treat the tissue first to what they call unmask the antigen, because the formalin fixation can possibly hide the epitope. And then you add the primary antibody, the secondary antibody, and the substrate, and then you see color. And I'm going to show you an example right here, again from Mary Hendrix's lab, the same paper in which they looked at the expression of that same protein nodal in normal breast and then in tumors of increasing aggressiveness. So this is a normal breast tissue, and now the brown color is nodal expression. So normal breast is not expressing nodal at all. If you look at ductal carcinoma in situ, this was kind of, John Licht explained this a little bit. So this is a tumor where the cells are confined to the duct and have not broken out of the duct yet. So this is considered to be a much less aggressive tumor. You're not seeing any nodal expression in this at all. If you look at an invasive ductal carcinoma where the tumor cells have now broken through the duct and into the surrounding supportive tissue, you can see now that there is some brown staining. Notice it's not uniform. And then if you look at a metastatic breast tumor, you can see that nodal expression is much more prevalent as noted by the brown color. So these are basically two ways to look at protein expression. Immunohistochemistry is very commonly used clinically to characterize tumors. For example, if you're looking to see is a breast tumor estrogen receptor positive or negative, you will often use immunohistochemistry to stain for the estrogen receptor expression.